Hello everyone, myself Triveni, Assistant Professor. In this video, I am going to explain cholinergic agents. Cholinergic agents, also known as parasympathomimetic agents. Here I am going to explain mainly what is agonist, what is antagonist and mixed agonist and antagonist, and biosynthesis of acetylcholine, metabolism of acetylcholine, and explanation with animation. So first of all, agonist. Agonist is a drug or agent that mimics the action of the signal ligand by binding or activating the receptor to produce biological response, that is known as agonist. Antagonist is a drug or agent that typically binds to a receptor without activating them, but instead decreases the receptor's ability to be activated by other agonist, which altogether it stops the receptor from producing a response, is known as antagonist. An agonist antagonist, also known as mixed agonist antagonist, is a drug or agent which under some conditions it behaves as an agonist, while under some conditions it behaves as an antagonist. When you observe this picture, this is agonist and this is the receptor. When agonist bind with the receptor undergoes some conformational changes, then it shows the turn on state. It shows biological response because of the fully activation of the receptor. When you observe antagonist, when antagonist bind with the receptor with the various mechanism, it produces turn off state. That means it does not produce the biological response. The receptor will not activate it to produce the biological response. One more is there, that is mixed agonist and antagonist. Here partial activation of the receptor will be taken place to produce partial biological action. This is regarding agonist, antagonist and mixed agonist and antagonist. Coming to the neurotransmitter. A neurotransmitter is a chemical messenger which is used to communicate in one direction from one neuron to another neuron, is known as neurotransmitter. Acetylcholine is a neurotransmitter which involved in the transmission of impulses from one neuron to another neuron or from neuron to other organs, is known as acetylcholine. This structure represents the acetylcholine. The left side it is an acetyl moiety and right side part is known as choline. This is the structure of acetylcholine. Coming to the neurotransmission. Here I will explain in brief about where acetylcholine released. Mainly acetylcholine works as neurotransmitter in peripheral nervous system. In case of somatic nervous system, acetylcholine released by the motor neurons at neuromuscular junction, which is present between the motor neuron and the skeletal muscle. In case of the autonomic nervous system, it does not matter about whether it is parasympathetic or sympathetic nervous system. The neurotransmitter released by the preganglionic neuron is always acetylcholine, but it is different for postganglionic fiber. In parasympathetic nervous system, acetylcholine is released by the postganglionic fiber, whereas noradrenaline will be released by the postganglionic fiber in the case of sympathetic nervous system. Finally, when acetylcholine binds with the receptors present on the adrenal medulla, it triggers the release of adrenaline in blood circulation. Now let's discuss the biosynthesis of acetylcholine. In the first step, coenzyme A is acetylated with acetate with the help of ATP in presence of the enzyme acetyl-CoA synthetase to form acetyl-CoA. At high glucose levels, acetyl-CoA is produced after glycolysis, in which final product of pyruvate undergoes oxidative decarboxylation to produce acetyl-CoA. Whereas at low glucose levels, acetyl-CoA can be synthesized by beta oxidation of fatty acids. Next one, choline is present in the cytoplasm, is transported into the nerve terminal by a specific carrier system, that is sodium dependent channel. Then this acetyl-CoA combines with choline to form acetylcholine. Acetylcholine acts as a neurotransmitter. This reaction was catalyzed by the choline acetyl transferase. During the reaction, acetyl group is transferred to choline, so the name of the enzyme is choline acetyl transferase. Let us see metabolism of acetylcholine. When we observe the structure of acetylcholine, it contains ester linkase. This ester linkase can be broken down or hydrolyzed with the help of esterase. So, acetylcholine in presence of acetylcholinesterase, it gets hydrolyzed into acetate and choline. This metabolism occurs at synapse. Let us see how acetylcholine is synthesized, stored, released and undergoes metabolism at ganglionic site. This is preganglionic nerve. This is postganglionic nerve, which has cholinergic receptors. The gap between preganglionic nerve and postganglionic nerve is known as synapse. Preganglionic nerve is embedded with choline transporters, through which choline is actively transported along with the sodium ions. This transport can be inhibited by hemicolonium. Once choline enters into the preganglionic nerve, it reacts with the acetyl-CoA in presence of choline acetyl transferase enzyme to form acetylcholine. Then synthesized acetylcholine transported into the presynaptic vesicles with the help of specific carrier proteins. This step can be inhibited by vasamicol. In the next step, when action potential reaches to nerve terminal, it opens the voltage gated calcium channel. Thereby, calcium enters into the nerve terminal. When sufficient concentration of calcium entered into the nerve terminal, the acetylcholine stored vesicle opened and releases the acetylcholine. This process is known as exocytosis. This neurotransmitter release can be inhibited by the botulinum toxins. Once acetylcholine released into the synapse, it can bind with the receptors to exert biological response. Free form of acetylcholine, which is present at synapse, can be metabolized by the acetylcholinesterase enzyme. This acetylcholinesterase enzyme, it metabolizes the acetylcholine and it forms the choline and acetate. This newly formed choline again reuptaken by the preganglionic nerve through choline transporters to synthesize another molecule of acetylcholine. Thanks for watching.